ಓಕೆ, ಈಗ ಹ್ಯೂಮನ್ ನರ್ವಸ್ ಸಿಸ್ಟಮ್ ಕಾಣ್ತಾ ಇದೆಯಲ್ಲ? ಹಲೋ. ಓಕೆ, ಈಗ ನಾವು ಲಾಸ್ಟ್ ಟೈಮ್ ನಿಲ್ಸಿದ್ರಿಂದ ಇದು ಮಾಡ್ಬೇಕಾ ಅಥವಾ ಲಾಸ್ಟ್ ಟೈಮ್ ಕೇಳ್ತಿದ್ದನ್ನ ಮತ್ತೆ ಸರಿಯಾಗಿ ಹೊಸದಾಗಿ ಮಾಡ್ಬೇಕಾ ಹೇಳಿ? ಲಾಸ್ಟ್ ಮೊನ್ನೆ ಸಾಟರ್ಡೆ ಸರಿ ಆಗಿಲ್ಲ, ಅದು ಏನ್ ಮಾಡ್ಬೇಕು? ಆಗಿದ್ಯಾ? ನರ್ವಸ್ ಸಿಸ್ಟಮ್ ಮುಂದೆ ಹೋಗ್ಲಾ? ಹಲೋ ಹಲೋ.
Hello, sir.
ಈಗ ಇದು ಟ್ರಾನ್ಸ್ಮಿಷನ್ ನರ್ವ್ ಇಂಪಲ್ಸಸ್ ಹೋಗೋಣ ಇವತ್ತು, ಅಥವಾ ಏನಾದ್ರೂ ಪ್ರಾಬ್ಲಮ್ ಇದೆಯಾ ಮೊನ್ನೆ ಹೇಳಿದ್ರಲ್ಲಿ? ಹೇಳಿ.
Sir,
ಹಾ,
sir, ಏನು ಕೇಳಿಸ್ಲಿಲ್ಲ ಆಗಿತ್ತು.
ಹಾಗಾದ್ರೆ ಹೊಸದಾಗಿಂದಾನೇ ಬರ್ಬೇಕಾ ಬ್ಯಾಕ್ ಆಗಿ? ಓಕೆ, ಐ ಡೋಂಟ್ ಮೈಂಡ್. ಸರಿ, ಹಾಗಾದ್ರೆ ವಿ ವಿಲ್ ಸ್ಟಾರ್ಟ್ ಇಟ್ ಫ್ರಮ್ ದ ವೆರಿ ಬಿಗಿನಿಂಗ್. ಓಕೆ, ಈಗ ಸ್ಕ್ರೀನ್ ಶೇರ್ ಆಗ್ತಾ ಇದೆ, ನಾನು ಹೇಳಿದ್ ಕೇಳ್ತಾ ಇದೆ ಅಲ್ಲ? ಓಕೆ.
Yes, sir.
Okay. Good morning to all of you. So I have to repeat what we have discussed in the previous class. So the nervous system that we are going to study is the most interesting and fascinating thing in any of the living beings. Probably you may be wondering, suppose if you touch any hot object, or suppose if you see some unwanted things, all of a sudden you show the reaction. For example, if you touch the hot object, within a fraction of a second you withdraw your hand. What makes you to withdraw your hand? The first important thing what you can see here is the very when you touch the hot object, that sense of heat is perceived by your brain, and that brain asks your muscles of the hand to withdraw it immediately. How much time it will take? It will in milliseconds. So now the question comes, how this quick reaction is made possible? So we can say in one word, this is made possible because of the human or the nervous system. So please switch on ನಿಮ್ದು ಮೈಕ್ರೋಫೋನ್ ಮ್ಯೂಟ್ ಮಾಡ್ಕೊಳ್ಳಿ, ಬಹಳ ನಾಯ್ಸಿ ಇದೆ ಇವತ್ತು. So this nervous system is the most advanced form. But whereas if you take in the primitive organisms, it started with some sensory cells. Then over a period of time, that got evolved into the most sophisticated, complicated nervous system in the mammals. Basically, when we talk about the nervous system, it includes two things. One is the central nervous system, which consists of brain and spinal cord. ನಿಮ್ದು ಮೈಕ್ರೋಫೋನ್ ಮ್ಯೂಟ್ ಮಾಡ್ಕೊಳ್ಳಿ, ಆಮೇಲೆ ಓಪನ್ ಮಾಡೋ ಅಂತ ಯಾರಾದ್ರೂ ಒಬ್ಬರು ಓಪನ್ ಮಾಡಿ ಇಟ್ಕೊಳ್ಳಿ, ಉಳಿದವ್ರು ಮ್ಯೂಟ್ ಮಾಡಿ ಪ್ಲೀಸ್. So central nervous system consists of two things, brain and the spinal cord. Then another part of the nervous system, what we call the peripheral nervous system, which goes to each and every part of the body. So here, first let me talk little bit about the central nervous system. Central nervous system, as I have told you, consists of two things. One is the brain, which consists of three regions: forebrain, midbrain, and hindbrain. I am not going to the details of the brain, as you have already studied that in the previous classes. All you have studied how there is a gyre, sulci, gray matter, white matter, all those things, convulsions and all those things. I am not going to detail. But another part of the central nervous system is the spinal cord, which starts from the medulla oblongata. In the previous photograph, you must have seen this is the medulla oblongata. From there, it runs downwards. So, extend downwards within the canal, which is found in the vertebral column. This spinal cord is also covered by three layers, as you have seen in case of the brain. That is outer durameter, middle arachnoid membrane, and the inner pia meter. There are three layers, and we call it as meninges. So there are three meninges: there is the durameter, outer durameter, middle arachnoid membrane, and inner pia meter. The length of the spinal cord is about 45 centimeters in male and 43 centimeters in case of the female. This is always, there's a height class difference in male and female, with though there may be some exception. But generally talking, the males have around 45 centimeters length of spinal cord, whereas the females have around 43 centimeters of spinal cord. And if you analyze the central cord, the central cavity is surrounded by inner gray matter and outer layer of white matter in case of the spinal cord. See here, you can see the spinal cord meninges or layers. So the innermost one is the pia meter. Followed surrounding that, there is another meninges, what you call arachnoid layer or arachnoid membrane, and outside we have dura meter. Here in this entire spinal cord, the photograph shows you there is an innervation of both red blood vessels, blood vessels, both arteries and veins, because they supply oxygen and nutrients to this spinal cord. If you see the spinal cord consists of totally there are 31 segments. Spinal cord consists of 31 segments. Here I have enlarged one segment, and from a in this spinal cord, each segment gives rise to one pair of nerves. Each spinal cord section gives rise to one pair of nerves, and each nerve has two roots. One is the ventral root and another one is the dorsal root. This ventral root and dorsal root together they form the spinal nerve, and this spinal cord cross section, if you see one segment, inner one is the gray matter.
ಸರ್ ಕಾಣಿಸ್ತಾ ಇಲ್ಲ.
ಮತ್ ಕಾಣಿಸ್ತಾ ಇಲ್ವಾ ನೋಡಿ, ಯಾವುದು ಕಾಣಿಸ್ತಾ ಇಲ್ವಾ? ಹಲೋ, ಎಲ್ಲಿ ತನಕ ಕಾಣಿಸ್ತಾ ಇತ್ತು ಇದು?
ಸರ್ ಅದೇ ಸ್ಲೈಡ್ ಇದೆ, ಟ್ರಾನ್ಸ್ಮಿಷನ್ ಆಫ್ ನರ್ವ್ ಇಂಪಲ್ಸ್.
ಚೇಂಜ್ ಆಗಿಲ್ವಾ?
ಇಲ್ಲ ಸರ್.
ಅರೆ, ಒನ್ ಮಿನಿಟ್ ಈಗ ಚೇಂಜ್ ಆಗ್ತಾ ಇಲ್ವಾ? ಹಲೋ, ಚೇಂಜ್ ಆಗ್ತಾ ಇದೆಯಾ ಇಲ್ವಾ ಹೇಳಿ? ಹಲೋ, ಇದೇ ಸ್ಲೈಡ್ ಇದೆಯಾ ಅಥವಾ ಚೇಂಜ್ ಆಗಿದೆಯಾ? ಹಲೋ ಹಲೋ, ಹಲೋ, ಕೇಳ್ತಾ ಇದೆಯಾ? ಕೇಳ್ತಾ ಇದೆಯಾ? ಹಲೋ, ಕಾಣ್ತಾ ಇದೆಯಮ್ಮ? ಸ್ಕ್ರೀನ್ ಕಾಣ್ತಾ ಇದೆಯಾ? ಹಲೋ,
ಇಲ್ಲ ಸರ್.
ಯಾವುದು ಕಾಣಿಸ್ತಾ ಇಲ್ಲ? ಏನ್
ಇಲ್ಲ ಸರ್.
ಸ್ಕ್ರೀನೇ ಕಾಣಿಸ್ತಾ ಇಲ್ಲ. ಓಕೆ, ಈಗ ವಿಸಿಬಲ್. ಈಗ ನಾನು ಸ್ಲೈಡ್ ಚೇಂಜ್ ಮಾಡಿದ್ರೆ ಚೇಂಜ್ ಆಗುತ್ತೋ ಅಂತ ನೋಡಿ, ಒಂದ್ ನಿಮಿಷ ಚೇಂಜ್ ಮಾಡ್ತೇನೆ. ಚೇಂಜ್ ಆಗತ್ತಾ?
ನೋ ಸರ್. ನೋ
ಅದೇ ಇರುತ್ತೆ, ಚೇಂಜ್ ಆಗೋದಿಲ್ಲ. ಈಗ ಸ್ಲೈಡ್ ಚೇಂಜ್ ಆಗೋದಿಲ್ವಾ? ಹಲೋ,
No, sir.
ಇಲ್ಲ, ಒಂದು ನಿಮಿಷ ಬೇರೆ ನೋಡಿ ಮಾಡಿ ನೋಡ್ತೇನೆ. ಈಗ ಈಗೇನಾದ್ರೂ ಕಾಣಿಸ್ತಾ?
Yes, sir.
ಈಗ ಏನ್ ಕಾಣಿಸ್ತಾ ಇದೆ?
Spinal cord, sir.
ಈಗ ಚೇಂಜ್ ಆಗುತ್ತಾ?
No, sir. Yes, sir. Yes, sir.
ಚೇಂಜ್ ಆಗ್ತದಲ್ಲ?
Yes.
Yes, sir.
So here I was talking about, see, this is the cross section of the spinal cord where you can see two nerves originating and forming a spinal nerve. That the gray matter, sorry, that the one is the ventral spinal root. Actually, this brings the information from different parts of the body, and this is the motor nerve. That is what you call the dorsal root of the spinal cord, which carry the information to other parts of the body. Now, in the spinal cord, the nerve bodies are located in the gray matter and nerve fibers are in the white matter. You know what is white matter and gray matter, which you have studied in the brain also. There are totally 31 pairs of spinal nerves, and this, irrespective of the size, though the length of the spinal cord may be more in the male and less in the female, but as far as the segments of the spinal cord are concerned, they are 31. That is fixed both for male as well as for the female. So 31 from each segment, a pair of nerve arises. As there are 31 segments, 31 pairs of spinal nerves arise from the spinal cord, and each nerve is formed by two roots, as I have told you. One is the dorsal root, which is the sensory nerve fiber, which bring the information, and another one is the ventral root, which is the motor nerve fiber. So now the spinal cord has, I have told you, there are 31 segments. What are these segments? There are 8 cervical segments that are found in your neck region. Then 12 in the thoracic segment, just behind the chest. In the vertebral column, you can see there are 12 segments. In the abdominal region, there are 5 segments, what you call lumbar segments, and 5 more in the sacral segment. And last one is the coccygeal segment, which is a tail. In case of the human being, it has been reduced. So altogether there are 31 segments, and these segments of the spinal cord corresponds to 31 pairs of nerves, because from each segment there arises a pair of nerves. See, this is the how the segments appear here. See, this is the 8 cervical, 12 thoracic, 5 lumbar, 5 sacral, and last one the coccygeal. Now, coming to the structure of a cell of the nerve cell. ಕೇಳ್ತಾ ಇದೆಯಲ್ಲ? ಹಲೋ, ಕ್ಯಾನ್ ಯು ಹಿಯರ್
Yes,
ಮೀ?
sir.
ಓಕೆ, ಸ್ಲೈಡ್ ಚೇಂಜ್ ಆಗ್ತಾ ಇದೆ.
Yes, sir. Yes, sir.
Okay. Now, the very basic unit of uh, nervous system is the neuron, or what you call the nerve cell. A nerve cell is a long filamentous cell, consists of two regions. One is the cyton and another one the axon. Cyton, the head, so-called head, which you can see, that is the nerve body, also called as. This cyton is having lot of mitochondria and a nucleus. The periphery of the cyton is drawn into number of branches. I hope you can see that. These branches are known as dendrites. Actually, these dendrites are responsible for receiving the information and passing on to the cyton. The information comes from different sources or different parts of the body, and that information that has been gathered by these dendrites, the hair-like or finger-like branches which you can see on the cyton, they are responsible for receiving the information from different parts of the body, and once this information in the form of electric impulses reach the cyton, there is some processing will be there. Then the same information is passed on to the next neuron. So for this reason, from the cyton, there arises a long fiber, and that is known as axon. This axon is a long fiber, and at the end of the axon is also branched. And but the difference is actually the tip of the axons. They are having buttons. I will explain and show you in the later when we discuss about the nerve conduction. First, that is in brief, this is the structure of the neuron. And this on the axon, you can see some beaded appearances. This is because of the protein coat. On the axon, you can, in most of the advanced nerve fiber, there is a protein coat known as myelin. This myelin coat is found in most of the neurons, but there are certain neurons which are free from this myelin coat. There is no myelin coat. If the axon is coated with the myelin protein, such neurons are known as myelinated neurons or medullated neurons. Again, I will repeat, if the axon is coated with a myelin protein, then such neurons are known as myelinated neurons or medullated neurons. And if it is absent, then it is known as non-myelinated or non-medullated neurons. Now you may ask, what is the function of this neuron coat? Actually, the presence of this myelin sheet on the axon enhances the or accelerate the nerve conduction. In any neuron which is having a myelin coat, that has been known to conduct the information or impulses very fast. If the myelin coat is not there, the conduction of the impulses is always very slow. I will come to later about how that works and all those things. Now, so this is another 3D image of the typical nerve cell or neuron. This is the cyton or cell body, what you call. These are all the dendrites from where it receive the information. Then the same information is passed through the axon. And see, on the axon there is a myelin coat, and that is not also continuous. The myelin coat is not continuous. There are interruptions. And at the tip, the axon forms again branches, and branches are ending with some synaptic buttons. This is in brief about the structure of a neuron. And in neurons, there are three different types: multipolar, bipolar, and non-polar. So in this multipolar neurons, see here the conducting zone, each one is having a conducting zone. It has more conducting zone, and it can transmit the impulses in different directions. Or so that's why the flow of information is in different direction. So that's why more than two direction, then you call it as multipolar. The second one, it is only bipolar. That means it can conduct the information from here to here or here to here in only two directions. So that's why it is known as bipolar. And last one is the monopolar neuron. And this monopolar can conduct the information unidirectionally. That is only in one direction. So based on the direction in which they can pass on the information, these neurons are classified into multipolar, bipolar, and non-polar reaction. Now, the very interesting fact here is how the nerves are able to conduct the impulses. All of us know now the neurons, the individual cells, or the basic units of this nervous system are responsible for the transmission. How they transmit? Actually, the neuron transmit the information in the form of electrical signals or electrical impulses. Once the electrical impulses are generated, the neurons have the capacity and capability to transmit to the target organ, and that's why the generation of an electric signal is very, very essential. A neuron is in a resting potential when these electrical impulses are minus 70 millivolts. Any change in the electrical signal, that means above minus 70 millivolt, it can create or it can uh, ignite this transmission of the impulses. So now what happens is that first, in order to generate the impulses, there a depolarization should be caused in the axon region, and thereby from resting potential nerve enters into action potential. So action resting potential is minus 70 millivolt, but above that it will be the action potential. Just give you an information, the resting potential. What do you mean by resting potential? The difference between the two potential charges, that is the positive and negative charges, is called the resting potential. And if it is minus 70 millivolt, then you call it as resting potential. The potential is the difference between the two potential charges, positive and negative. And the membrane of a neuron, if you see, it's very interesting. The membrane of a neuron is negatively charged internally and positively charged out externally. So the same membrane, internally it is negatively charged and externally it is positively charged. This generates a potential difference of minus 70 to minus 90 millivolts. See here, this is the section of the axon. See, internally this is the neuron. Internally the membrane is having negative charges. Externally outside it is having the positive charges. The difference is almost as I have told you, the about minus 50 to minus 90, average is minus 70 is the resting potential. If the because of the potential difference, if the electric voltage generated is in between minus 50 to minus 90 millivolts, then you can say that the neuron is in resting potential. It is not conducting any information. Now you look at it. In order to how this resting potential is maintained. So here there are certain pumps, cation pumps. Sodium pumps maintain the active transport of potassium ions in and sodium ions out of the neuron. So there are certain pumps, especially the sodium pumps. They are responsible for maintaining an active transport. Active transport means with the expenditure of energy of potassium ions inside. They send the potassium ions inside and send the sodium ion outside. When three sodium ions are pumped out, two potassium ions are pumped in. See, just you see the difference. Both are positively charged, sodium and potassium. And when three sodium ions are pumped out, only two potassium ions enter inside through this sodium pump. And of course, this is generally known as sodium potassium ATPase pump because it is ATP is the energy currency and it requires energy. So that's why it's an active transport where there is a utilization of adenosine triphosphate. So that's why when three sodium ions are pumped out, only two sodium potassium ions come inside. See here, this is the actively transport. See this is the membrane of the nerve cell. See, this is the sodium potassium ATPase pump, what we call. When three sodium ions are pumped outside, only two potassium ions are pumped in. This is outside the cell and this is the inside the cell. I hope it's clear now. See, this is also another sodium potassium pump photograph. When three sodium ions are pumped outside, two potassium ions are pumped inside. And see, here inside the negative charge is maintained and outside the positive charge is maintained, which you can always see there. Then in spite of this, there is a movement of the cations, sodium and potassium. So Also within the membrane there are channel proteins, and these channel proteins which I have just shown you allow both sodium and potassium ion diffuse back down their concentration gradient. Now, with the help of active transport, what has been done is 3 sodium ions are sent out, 2 potassium ions are brought in. But in addition to the sodium potassium ATPase pump, which function against the concentration gradient with the expenditure of energy. But here there are many protein channels also. These protein channels are meant for passive transport that allow the sodium and potassium ions to diffuse back down their concentration gradient from the region of higher concentration to the region of lower concentration. But what the problem here is there are many more potassium channels and less sodium channels. So that's why more potassium channels diffuse back. They go outside easily, but the sodium ions from outside, they cannot come that much easily inside because the protein channels for potassium are more and for sodium they are less. So more easily and more number of potassium ions, they diffuse back, they go outside. But whereas same number of sodium cannot come inside as there are less number of sodium protein channels. As a result, what has happened outside is that the outside of the axon is positively charged compared to inside. So, in the nerve membrane, outside is more positively charged compared to inside because there will be more and more sodium ions are going, but the same number of potassium ions are not coming inside. Now, inside the axon, around 9 minus 70 millivolt, this is called as resting potential, which I have already told you. So, a nerve to change into from resting potential to action potential, the electrical charge should vary from minus 70 millivolt to plus 40 millivolt. And this condition is said to be membrane is depolarized. And action potential is the term when the nerve is able to conduct the impulses. Action potential is produced when the membranes of the neuron get stimulated and the charges reverse. The entire charges reverse. That means the electrical charges change from minus 70 millivolts to plus 40 millivolts. See, this is the action potential. This is the resting potential here you can see. Then all of a sudden there is a change in the because of the impulses, the electrical charges minus 70 millivolt to plus 40 millivolts, all of a sudden the nerve gets excited, and that is what you call the action potential. So how the action potential is generated? When you ignite the nerve impulse can be initiated either by mechanically, chemically, thermally, electrical stimulation, anything. Even if you touch it, how do you feel that? An action potential is created and the feel of the touch is conveyed to your brain, and thereby you experience the feel of touch. So the nerve impulse can be initiated by mechanically or chemically or by heat or by other electrical stimulation. What should happen is when the nerve is in resting potential, that is the minus 70 millivolt, it should change to plus 40 millivolt. This change in the potential and making the impulse to be carried, that is known as action potential. See here the action potential where you can see what is happening. There's a change in the cations and anions. I have told you inside it is negatively charged, outside it is positively charged when it is in a resting potential. But when the action potential starts, there is a reversal of the charges. Inside becomes positively charged and outside becomes negatively charged. And that area of the nerve is said to have in an action potential. Now what has happened, I will show you now. See here also. The resting potential, immediately the sodium ions began moving in and the continuity of the moving will speed up, and as a result there will be difference in the charges. Positive becomes negative and negative becomes positive. As a result there will be net movement of the ions and that will show you the action potential. How the action potential is produced when there's a sudden increase in the permeability of the membrane to the sodium ions, and sodium ions rush into the neuron through sodium channels and thereby they depolarize the membrane and then further increases permeability to sodium. First, sodium ions rush into the neuron through sodium channels and there they depolarize the membrane. Once the membrane depolarized, it will further increase its permeability to sodium ions. So as a result, this leads to a greater influx and further depolarization. So the sodium ions move into the axon, causing a change in the electrical charges from minus 70 millivolts to plus 40 millivolts. This reversal, minus 70 to plus 40 charges, causes this action potential. Now, when inside becomes sufficiently positively charged because of the flow of the sodium ions, permeability of the sodium ions decreases. First, there's an increase in the permeability of the sodium ions inside. But once the membrane is sufficiently positively charged, the permeability to the sodium ions start decreases. At the same time, sodium ions begin to move inside and potassium ions begin to move in the opposite direction. Along the diffusion gradient or along the concentration gradient without the expenditure of energy. Slowly what has happened, this electrical potential from plus 40 it comes to minus 70 and that is what we call diffuse slowly until the membrane is repolarized from depolarization to it has come to depolarization, and all these things happen within 2 milliseconds. Same portion of the membrane returns to the within 2 milliseconds, the same portion of the membrane returns to the resting potential, that is minus 70. See how much time it requires to change from minus 70 to plus 70 and plus 40 and again coming back to minus 70, 2 milliseconds. And this is what we call repolarization, that is from plus 40 milliseconds to minus 70 millivolts plus 40 millivolts to minus 70 millivolts. It takes hardly any 2 milliseconds, and once it comes there the neuron is said to be repolarized and now we call it as resting potential. So stimulus exceeds a certain value, then only it can uh, cause this action potential. And here, above the threshold value, what do you mean by threshold value? See, in the earlier one or two, this thing I must have shown you. This is, I can show you one photo. See, this is the threshold value. Minus 60, minus 70 is the resting potential. And even if it comes to 40 milliseconds also nothing is shown. That is the, and above minus 40 slowly the action potential starts. So this gap minus 60 to minus 40, this is the threshold level. Only if your stimulus is strong enough to generate an electrical power, electrical current more than minus 40 millivolts, then only the action potential starts, and that is known as threshold level. So here there's one principle, all or none response. Above the threshold value, the size of the action potential remains constant. So it doesn't mean that regardless of the size of the stimulus, even if you above the threshold level, the information or the impulses that is carried remains same and the action potential also remains constant irrespective of the stimulus. The size of this action potential does not decrease as it is transmitted along the neuron but always remains the same. And the size of the action potential or the intensity of the action potential will not decrease as it moves from one part of the neuron to other part of the neuron. Same is maintained. Progression of impulses. You see, when a nerve impulse reaches any point on the axon, the action potential is generated, but this impulse, the intensity of the impulse will remain same. Small local circuit exit at the leading edge of the action potential, and sodium ions move toward the negatively charged charges. So that's why the impulses has been formed. This excites the next part of the neuron, and so that the action potential progresses. That's the one photograph I'm searching for that. Uh, absolute refractory, two terminologies I would like to introduce to you. One is absolute refractory period. This lasts for 1 millisecond during which no impulse can be propagated however intense the stimulus is. What does it mean? It's a duration of very short period, 1 milliseconds. After one action potential, it requires a gap of 1 milliseconds for the generation of next action potential. You cannot avoid this lag, however intense the stimulus may be. Stimulus may be, hello, can you hear me? ಕೇಳ್ತಾ ಇದೆಯಾ? ಹಲೋ, ಕೇಳ್ತಾ ಇದೆಯಾ ಅಥವಾ ಇಲ್ವಾ? ಹಲೋ, ಕೇಳ್ತಾ ಇದೆಯಾ? ಸಾರಿ, ನಾವು ಹಲೋ ಕೇಳ್ತಾ ಇದೆಯಾ ಅಥವಾ ಕಟ್ ಆಯ್ತಾ? ದೇರ್ಸ್ ಅನ್ ಇಂಟರ್ನೆಟ್
Yes,
ಪ್ರಾಬ್ಲಮ್.
sir.
ಏನ್ ಕೇಳ್ತಾ ಇದೆಯಾ?
Yes, sir.
ಓಕೆ, ದೇರ್ ಇಸ್ ಸಮ್ ಇಂಟರ್ನೆಟ್ ಏನೋ ರಿಪೇರ್ ನಡೀತಾ ಇದೆ. ಫಾರ್ ದ ಟೈಮಿಂಗ್ ಐ ವಿಲ್ ಸ್ಟಾಪ್ ಅಂಡ್ ಐ ವಿಲ್ ಕಂಟಿನ್ಯೂ ಟುಮಾರೋ. ಓಕೆ, ಈಗ
Okay,
ಇಂಟರ್ನೆಟ್ ನೆಟ್ ಕನೆಕ್ಷನ್ ಹೋಗ್ತಾ ಇದೆ, ಸೊ ಐ ಹ್ಯಾವ್ ಟು ಲೀವ್ ದ ಮೀಟಿಂಗ್. ಓಕೆ,
okay, sir.
Okay, sir.